In this video, what I'm going to be going through are the specific issues that have to do with gene cloning. If you haven't had a look at the social, biological, and ethical issues video that explains what each one of those ones is, go back and watch that one before coming and watching this one. So the study design dot point says this. It says you need to understand the technique of gene cloning. They've taken out genetic screening and DNA profiling. You also need to understand social and ethical implications and issues. So the biological issues aren't really in here, but we will go through them anyway because it's nice to look at them on the whole. So before I start to go through them, you might want to think about and start to make a list. You can do it on a mini whiteboard, you can do it on a piece of paper, you can pause, think, what are some of the biological issues to do with gene cloning, social issues and ethical issues? What have we got that we are dealing with these? And hopefully you pause and then you come back. And you come back. So here's a little table and I'm just going to go through each one bit at a time. So you can see up the top here, these are some of our social implications in blue. We get, again, here's something to do with costs. So if you make therapeutics with bacteria, so this would be e.g. insulin, some of the benefits to that are the fact that it doesn't cost as much to use bacteria to make insulin versus pigs. We also get wider access to treatments because we make them cheaper, so therefore they can go further in society. So more people can afford them. More people afford. I can't write today, but that's okay. We also get more employment opportunities. So anything to do with employment is going to have a societal benefit. We often see at the moment on the news, they're talking about unemployment rates rising. So if you could create jobs in a biotechnology industry, then you're going to be benefiting society. So this one here is definitely a plus. We've got wider access to treatment is a plus. Reduced costs is a plus. Now the last one here is the misuse of this technology to create things like performance enhancing or illegal drugs. So if you create these illegal drugs using this technology, they might not be able to be detected by standard tests. So therefore you can get away with it. This would be what I would call a negative implication on this issue. So we had 3 positives of gene cloning and 1 negative just in this table. Now this list is not exhaustive. If you can think of other examples that might impact society, put them down, run them by me, not a problem. So let's go on to the next one and look at biological implications. So here is one example. So they've got these human recombinant proteins are more effective than proteins purified from other animals. So for example, you make insulin from bacteria is better and it actually works in our bodies more effectively than the stuff that we extract from animals. So that's definitely a plus. You also, though, when you do bacterial transformation, bacteria are small. So when you are extracting and purifying your insulin, say, there's always a risk of contamination. Yeah, and that contamination could be introducing bacteria into your insulin injection, then you're injecting that bacteria into a human, and that could have implications. Now, we have lots of checks and balances that stop that from being a big issue, but it's still an issue. Another biological implication is that we can actually use gene cloning to have newer and safer vaccines, also newer testing procedures. Use gene cloning, like the COVID test uses PCR. So all of these and that's gene cloning. So we are cloning a gene over and over again, and that is enabling us to detect whether or not a person is infected with the coronavirus. So that's definitely a positive there. So the final one that we're going to talk about with gene cloning are the ethical implications. So these are the ones that are morally kind of ambiguous, whether or not you believe they are right or you believe that they are wrong. So this says the use of bacteria for therapeutics addresses philosophical, cultural, or religious objections to animal use. So what that is saying, that is a plus. Yeah. So it's saying that by using bacteria we are going around that cultural or philosophical animal use. So these are people like vegans and vegetarians are quite happy to use bacteria. So that's definitely a plus, that ethically for them, obtaining insulin from a bacterium is a lot better than obtaining insulin from a dead pig or cow. The last one is the major issue. If you ever are at loss for an ethical issue, you go with this one. Essentially, the manipulation of shifting of genes is unethical regardless of purpose. The phrase that you will see and the phrase that you can write is the fact that people believe that any manipulation of a gene is unethical. It's against what the natural way is, and so therefore you can say it's like playing God. What I would love for you to do is this little task. There's some pictures in your notes on the bottom of page 2. Have a look through this. Essentially, this is one application of gene cloning, and it's nice to have an application written down in your books. So it's all got to do with the fact that our body makes a protein called erythropoietin, and what it does is it stimulates the red bone marrow to produce more red blood cells, and it keeps us at a homeostatic level. So the kidney releases that erythropoietin. Problem is, if you start to get damaged kidneys, then you might not make as many red blood cells and you get this imbalance. And that's not great. If you get less red blood cells, you're going to end up with less oxygen carrying capacity. Yeah, so that's not good. So what we can do is we can actually take out the human gene for erythropoietin, put it into a recombinant plasmid, put that recombinant plasmid into a culture with mammalian cells. So essentially, we add them together with bacteria as well. Then we end up producing more of this human protein, and you can purify that and inject that into a human. So it's very similar to insulin, but this time instead of removing the human insulin gene, we're removing the human gene for erythropoietin. And that's just one example of how we use gene cloning.